Well, we're here today to do some survey work. We're working with Plymouth Marine Laboratory and we're here on their research vessel Quest. What we're going to be doing is taking water samples today to measure the productivity in the water column, the numbers and the diversity of phytoplankton that live in the water. These are the base of the marine food chain, so they're very important. And during the spring and the summer we get blooms, so increases in the abundance of these phytoplankton. So yeah, what we're measuring today is a range of different species and their abundances, and we're looking in particular for species of microscopic algae that make up the phytoplankton that are actually harmful. We call them harmful algal blooms. They're conducting a kind of biochemical warfare with one another to try and, dominate the water column. The trouble is, these toxins are taken up by animals that consume the plankton, like filter feeding shellfish, and of course then these toxins can concentrate in the shellfish and it really is then unsafe to eat. This is a problem. This is a big problem for the fishing industry, for the aquaculture industry. The toxin build-up in the shellfish means that those shell fisheries have to close, and this can have a big financial impact on the industry. And of course, here in the southwest, this is a big part of the industry that we have. The aim of our research is to really understand the environmental conditions that are necessary to promote these harmful algal blooms. If we can identify those factors, then we can start to be more predictive about when these events might happen. We're working with Plymouth Marine Lab to understand more, in this case, about a particular site of Plymouth. We're now motoring out to our first sampling station, which is quite close into the shore. It seems that the frequency of harmful algal blooms is increasing, and it appears to be increasing in line with climate change. These harmful algae prefer warmer temperatures. Using this Rosette water bottle sampling system, essentially a range of different bottles that close at different depths in the water column, capturing a sample of water, each of them 10 litres in volume, put them into sampling bottles like this, which can then be analysed back in the lab. This will contain a little water sample that will go for nitrogen analysis. Nitrogen and phosphate are essential nutrients for phytoplankton growth. Trying to link the abundance of these harmful algae with different environmental parameters. Variations in temperature in the water. This device is also loaded with a series of probes that can measure directly salinity, temperature, light attenuation, various different things. So we can build up a profile of the structure of the water column and also its chemical content. Today we're sampling again phytoplankton and water, but we're in the Kingsbridge estuary, Kingsbridge and Salcombe estuary. The seawater comes in on the high tide and it pushes the algae up to the top of the estuary, into the fingers of the estuary, and it's here where we get concentrations sometimes of harmful algal blooms. So we're again looking for the environmental conditions that promote these blooms in this particular area.
Right, so this instrument is a spectrophotometer and it allows us to look at cell density through all the amount of pigments that might be in the cell, so for example chlorophyll.
So if we can identify areas that are outside these harmful algal bloom hotspots, then we'll target those. It could be also that just identifying periods in the growing season when these blooms are more likely to happen, predicting when they're going to happen and forewarning shellfish operators so that they can perhaps harvest their stock early and avoid been contaminated by the blooms. So it's really about identifying where and when these harmful blooms are likely to take place.